you all. Now we are going live on YouTube. Okay, so currently we are live on YouTube. Welcome you all for an hour with an expert lecture series 3. Over to Dr. Dande.
So good afternoon. Welcome everybody. So this is the third in series, and we have a very good number of participants so far. Trying to listen to Dr. Deepak. I'll be introducing him later. Now let me hand it over to Dr. Manoj to take care of the proceedings.
So good afternoon, everyone. Let's start the proceeding. I will invite Dr. Raju Thomas, President, Ayurveda Medical Association of India, for welcome again.
Thank you, Dr. Manoj. Am I audible?
Yeah.
Good afternoon, one and all. At the very outset, I welcome all the dignitaries participating in this webinar from various parts of the globe. And I congratulate Mrs. Ultimate Pharmaceuticals and Ayurveda Vilasani for joining with the Ayurveda Medical Association of India for this webinar series, an hour with an expert. In the present context of the pandemic COVID-19, the subjects we selected are very important. In the current situation, Ayurveda has a lot of things to deliver, and a lot of people across the globe is keenly watching the Ayurveda sector. We should be able to communicate the Ayurvedic concepts, our principles, and the way our system works in an acceptable manner to the scientific community as well as the common man. I think these programs will help us to go long distances. This is the third lecture in this series, and eminent personalities from across the globe are delivering that. Today, we have Professor Deepak Ramji from Cardiff University as our faculty. I wholeheartedly welcome Dr. Deepak Ramji. I welcome Dr. Shiban Ganju, Chairman, Ultimate Pharmaceuticals, and Dr. C.V. Jayadevan, Principal, Kotak Ayurveda College, Dr. Rishikesh Damle, Managing Director, Ultimate Pharmaceuticals, Dr. Manoj Kalur, our member, former CCIM member, and Managing Director of the Ayurveda Vilasani Vaidyashala are also with us. I wholeheartedly welcome the dignitaries. General Secretary of Ayurveda Medical Association of India, Dr. Saad Dinagar, Dr. V.G. Udayakumar, Research Foundation Chairman, Dr. G. Vinod Kumar, President of AMAI, Dr. Leda Damle, Dr. Shaiju Pallakot, medical officer and researcher are also present with us. I wholeheartedly welcome all of you. I once again welcome all the dignitaries participating in this webinar from various locations. Thank you.
Welcome Dr. Rishikesh Damle to introduce the expert.
Dr. Ramji, Deepak Ramji was in our office a few months back, and if you just meet him for 10 minutes, you will know the depth of knowledge what he has. And we are very thankful to have him here giving us the lecture today. Dr. Deepak Ramji has done his BSc from Leeds University. Then he did PhD in biochemistry from University of Leeds. As of today, he is a professor of cardiovascular science, School of Biomedicine, Cardiff University. He was a lecturer and senior lecturer and reader at School of Bioscience, Cardiff University earlier till 2017. He was also a postdoctoral research associate with Professor Gennaro Gilberto at IRBM Rome. So he's basically an academic person. His research interests are basically understanding the molecular mechanisms between inflammation, immunity, atherosclerosis, and cardiac disorders in short. And this is what he's going to talk about today. He has acquired grants up to 4.3 million pounds for university and for various projects so far. He has published around 170 research articles. He was the principal supervisor of 23 PhDs, 2 MPhils, and 3 MREs that have been completed so far, and he is still guides a few PhDs. He was an expert evaluator and panel member for Horizon Panel Member Chair for Assessment Committee of IRC SCT Postdoctoral Fellowship Scheme. So he's regularly invited across the world for various talks, and the reason he had come to India was also to give his talk in University of Mangalore. So he has recently published several papers, and most of the papers are about inflammation and cytokines and atherosclerosis. So he'll be enlightening us about the same topic today, including immunomodulation. So I request Dr. Deepak Ramji to welcome you here, and it will be a pleasure to listen to you.
Okay, thank you, Rishikesh, for that kind introduction. Thank you also to Manoj, Raju, and the organizing committees. Now, can you see my slides?
Sir, we request you to share the slide now as share content.
Right, let me go back. Right, so that's that, okay. Right, can you see that?
Yes, perfect, sir.
Okay. So, thank you, obviously, for that kind introduction. It's also a pleasure for me to be giving this talk, this webinar, on what's actually a National Teachers' Day in India, in fact. And so, before I just sort of start talking about science, I would like to sort of very briefly talk about a little bit about my educational journey in a couple of minutes, because it is extremely interesting and full of ups and downs. And it's actually been inspirational to several younger sort of researchers that actually have heard about this. And I'm a firm believer that if, irrespective of where you start from, if there's a will, in fact, a way will be there and doors will open. So just to actually tell you in the context, I was born in this very small village in Uganda called Saroti. My father moved there from India when he was 10 years old. And Saroti, in fact, is a very small village. One of the attraction there was this hill, in fact, that you see, a picnic spot. And to just give you a context in terms of how small the village was, around sort of the 70s, there were only 10 television sets in the whole village. So it just gives you a little bit of context. Education only went up to the age of 16. So if you wanted to go beyond that, you had to go to the bigger cities or elsewhere. And so that's where I started my primary education. And in fact, as I was reaching, in fact, the middle stage of my primary education, my two elder brothers actually were elsewhere, getting out for the studies. My parents actually had this corner shop that you see here, and there's a mango tree next to this corner shop. It's still there. I actually spent a lot of time playing marbles and the Indian game Gilli Danda with a lot of my friends there. Now, of course, things changed drastically in August 1972 when the president there, Idi Amin, announced that all British nationals had to leave the country in 90 days. So what I found around that phase from August to December is that I lost virtually all my friends in a space of a couple of months. We stayed on because we're Ugandan citizens. And at that stage, of course, the family was split up because the elder brothers were in either in Kampala or in another country, Tanzania, later on, in fact, moving to Canada. And the reason my father sort of stayed on, because we're Ugandan citizens, and he thought, well, we will not be affected. And that didn't turn out to be the case. One day, the people from the army came along, took away the shop, and a week later, also the house. So within, in fact, a week, we met homeless. And the doctor there, in fact, the local doctor came out to rescue, to help out. And at that point, 4 of the members of the family, out of 6, 4 in fact, which was me, my sister, and my parents, moved to Baroda in India, where my maternal grandparents were. And so I then sort of tried to continue my education in Baroda. it wasn't really an education. You actually sort of found me more in cinemas. And in 4 years that I was in India, I actually sort of must have watched in excess of 500 Bollywood movies. So study, in fact, was completely out of question. And obviously, in fact, India was really wonderful in that sense. And a lot of things which I acquired in India is something which I continue in the UK as community service. I do a lot of community service in UK. One of them, in fact, is the so-called monthly Samu Pratna, and which we started in April. And Lata Ben Damle, in fact, joined us, in fact, singing a couple of bhajans in our first Samu Pratna in April. In late 70s, moved to Leeds, and that's where I really started my education. And for the next 4 years, I didn't see a single Bollywood movie. And so I did completed my secondary education there. My interest was in biochemistry and, my interest was in chemistry and biology. And so that was a natural route for a degree in biochemistry at Leeds University. I then stayed on in Leeds to do my PhD, and the PhD was on gene expression during development. And when I finished my PhD, the climate in terms of jobs in education was not that great. So I decided to move out of UK. I got a fellowship from the Royal Society and went on to a premier European molecular biology laboratories in Heidelberg, where I got interested in liver-specific gene expression. And this is, in fact, the EMBR laboratories in Heidelberg. And this was funded by the Royal Society Fellowship. Then I actually moved and did another research fellowship in Rome, in a sort of institute funded by Merck, Sharp, and Dolme. There was a little bit of introduction to industrial funding. My sort of fellowship was funded by the EU, and that's where I got interested in inflammation, liver inflammation during various diseases. And in 1992, I moved from Rome to Cardiff in Wales. This is Cardiff University. I started as a junior lecturer in biochemistry, and I've been there for 28 years, progressing from a lecturer to a senior lecturer, reader, and then, of course, a personal chair, which is actually a distinguished professorship. And so I've continued this theme on inflammation in Cardiff, starting with the liver. And then in the last 15 odd years, we're focused on atherosclerosis. And atherosclerosis, as you all know, is an inflammatory disorder of medium and large arteries that is associated with buildup of lipids and fats in the walls of the arteries. It's a chronic inflammatory disorder. And what it does is, in the mild form, causes narrowing of the arteries and restricts blood flow. The clinical complications arises when so-called atherosclerotic plaques rupture, and depending on the artery that is being affected, they will give rise to cardiovascular diseases such as myocardial infarction, heart attacks, cerebrovascular accidents, stroke, and peripheral vascular disease. Now, it's clearly a major killer worldwide. In the UK, it's responsible for around 25% of all deaths, 1 in 4 deaths in the UK. That is actually reduced in the last few years, in part due to lifestyle changes, in part due to pharmaceutical intervention. But still, it is a major economic burden, costing around 9 billion pounds to the healthcare in the UK every year. Now, of course, whilst as I mentioned, there's been reduction in morbidity and mortality from atherosclerosis and cardiovascular disease in the West, there is, in fact, a disturbing increase in sort of emerging economies. And India was one example. China is another one. And globally, it accounts for around 31% of all deaths. If you look at India, what you find is that whilst we don't have sort of full statistics, what we actually can gauge from various sort of surveys and so on, that in 2016 it accounted for around 28% of all deaths. It's around 33% now, and that's probably an underestimate. What is even more disturbing is if you look at trends from 1990s to 2010, when of course there's been a reduction in the West. In India, it's increased by almost 60%. And what also, in fact, is also quite alarming is that the onset is early compared to the West. It's an accelerated buildup, and there's a high rate of mortality, particularly, in fact, in individuals who are actually in the 30s and 40s. That is actually sort of changing, in fact, so I've been visiting India in the last 6 or 7 years on a yearly basis. And I see, in fact, the younger generation is actually becoming more active. But still, in fact, the incidence of risk factors associated with atherosclerosis, such as high levels of blood cholesterol, hypertension, and particularly diabetes, is quite ripe in the Indian population. And here, in fact, is a little bit of link with my interest in Bollywood. We have lost a lot of Bollywood stars because of atherosclerosis and heart attacks. So from the singer, we've got Mukesh, Muhammad Rafi, and Kishore Kumar. And then, of course, from the actors, several actors, Vinod Mehra is one of them, Navin Nishchal. And because I'm a Gujarati, my favorite one, of course, is Haribhai or Sanjeev Kumar. Interestingly, in fact, on Independence Day, the movie Sholay celebrated 45 years. And obviously, Sanjeev Kumar had the major say in that movie with the final dialogue, Ye haath mujhe de de gabbar. So clearly, phenomenal stars in the Bollywood industry, the singers, who actually unfortunately died prematurely because of atherosclerosis and its complications. So what I would like to do initially is run through the pathogenesis of this disease, because it will actually be extremely useful with some of the data that I'll be presenting later on in my talk. So the disease is initiated by various risk factors, and one of the key risk factors is accumulation of LDL cholesterol in the plasma. And this LDL will then move, in fact, through the walls of the arteries, the endothelial layer of the arteries, into the subendothelial space, where under condition of oxidative stress, the LDL will get modified. And one common modification is oxidation of LDL, so the LDL gets oxidized. And this actually then causes what's called endothelial cell dysfunction. And what happens in endothelial cell dysfunction is that the endothelial cells of the arteries will secrete chemo-attracting cytokines into the blood. These are called chemokines. And they will also increase the expression of so-called adhesion proteins on their cell surface, which act as glues. And so the chemokines will then attract immune cells, particularly monocytes, and this will attach to the adhesion proteins and then move into the subendothelial space. And so chemokine-driven monocytic migration is the second event in the pathogenesis of atherosclerosis. What happens is that as these monocytes which are recruited, they will then differentiate into macrophages. And this differentiation of monocytes into macrophages is associated with increased expression of so-called pattern recognition receptors on their cell surface. And of course, we all know that macrophages are key players in the innate immune system. And what these pattern recognition receptors recognize is signatures or patterns associated with pathogens and aid in the clearance of pathogens. Unfortunately, some of these pattern recognition receptors, so-called scavenger receptors also recognize signatures in modified or oxidized LDL. And so they'll bind to this receptor, and then via receptor-mediated endocytosis and also some of the other processes, the macrophages will take up modified LDL and transform into lipid-loaded foam cells. And these lipid-loaded foam cells form the fatty streak that you see during the early stage of the disease. So macrophage uptake of modified lipoproteins and transformation into lipid-loaded foam cells is an early event in the disease. Accumulation of cholesterol is toxic to cells such as macrophages. And so what will happen is that these macrophages will die via apoptosis and necrosis and deposit their lipid into the environment to form this lipid-rich necrotic core. And the lipids, which include cholesterols, cholesterol crystals, and other lipids, will then cause a state of chronic inflammation. And this is because activation of a lot of inflammatory pathways which are mediated by the action of cytokines. And one such pathway is activation of the inflammasome. And so what you reach here is an intermediate lesion. But because of this chronic inflammation, you have an atherosclerotic plaque that is prone to rupture. And so to counteract this as a more protective mechanism, the normally quiescent smooth muscle cells migrate from the media to the intima and lay an extracellular matrix which forms this plaque stabilizing fibrous scarp. So smooth muscle migration and fibrous scarp is an event that occurs later on in atherosclerosis when you go development of atheroma. What you reach now is a situation where there's a balance in the synthesis of extracellular matrix by smooth muscle cells and the degradation of this extracellular matrix by proteases which are produced under inflammatory condition. And during the chronic inflammation, you find that the synthesis of the fibrous scarp is inhibited, whereas its degradation is stimulated. And so excessive degradation will cause rupture of the plaques, thrombosis, and various clinical complications. Now, of course, this is the pathogenesis of the disease, which is quite useful for in terms of explaining data which we've sort of generated from some of our studies. It's worth also bearing in mind that COVID-19 also affects a lot of these steps as well, such as endothelial cell dysfunction, inflammation via the cytokine storm, and also, in fact, increasing platelet aggregation. And so the lethality of COVID-19 is much higher in individuals who have got relatively advanced atherosclerosis. So what we have done in the last 15 odd years is developed a lot of assays, in vitro assays that recapitulate various steps in the pathogenesis of the disease with multiple cell types, and this include monocytic migration, foam cell formation, and metabolic perturbations that you see associated with this disease. We also develop a range of in vivo preclinical model systems. This could include wild-type mice, which are fed a high-fat diet, and there are also metabolic changes that you see in the early stages of the disease, particularly risk factors associated with atherosclerosis. Now, wild-type mice normally don't develop atherosclerosis. So we have to use genetically modified mice and deficient in the LDL receptor, and mice which are deficient in apolipoprotein E, which is an apolipoprotein. And again, you have to feed these mice a high-fat diet. Now, just to actually mention one point is that obviously the cholesterol is carried, the bad cholesterol is carried in the blood by LDL. And the LDL receptor are involved in clearance of this cholesterol. And of course, Brown and Goldstein, who discovered the LDL receptor, won a Nobel Prize for that. And this explains why LDL receptor deficient mice develop atherosclerosis when you feed them a high-fat diet. It's worth mentioning that the LDL receptor per se is not a major contributor to atherosclerosis development, because the expression of the LDL receptor is under negative feedback regulation. So the more the cholesterol is taken by the LDL receptor, the less the LDL receptors are expressed on the cell surface. So they cannot be a huge contributor, at least in fact mechanistically, to atherosclerosis, sort of the latter stage of atherosclerosis development. On the other hand, the scavenger receptors take up LDL and modified LDL in an uncontrolled manner. So they are not under negative feedback regulation. And so in our in vivo models, we have a number of readouts, which includes also hematopoietic stem and progenitor cells in the bone marrow, because these have been shown to change also drastically during the pathogenesis of the disease. So here, in fact, is some readouts that we take using our in vivo mouse model systems, where we can look at immunoprofiling and lipoprotein profiling of peripheral blood. We can look at plaque burden in arteries. We can look at hematopoietic stem and progenitor cells in the bone marrow. We can do single-cell RNA sequencing. We can look at other organs which also undergo metabolic perturbations and contribute to atherosclerosis, such as the liver. And also, in fact, we can not only look at plaque progression, we can also look at plaque regression. So this, in fact, are development of plaques in mice and then changing diet, because regression of plaques and asking the question, do our agents actually cause more regression compared to a simple switch in diet from a high-fat diet to a chow diet. So that leads me quite nicely to talk about current therapies and their limitations. The majority of current therapies target the high cholesterol and lipid dysfunction or in lipid homeostasis that is associated with this disease. And one of the gold standards in terms of treatment are the statins family of drugs. Now, statins inhibit a rate-limiting step in the biosynthesis of cholesterol, and 75% of cholesterol is made by your body in the liver mainly. And so statins will inhibit the synthesis of cholesterol. And they have been extremely effective in reducing mortality and morbidity associated with atherosclerosis and its complications. And they have been described as blockbuster drugs. However, statin therapy is also associated with a considerable residual risk for cardiovascular disease. If you actually look at individuals who are on statins, 70% of those will still go on and have a heart attack, and that's because of this marked residual cardiovascular risk associated with statins. Not only that, there are a number of risk factors number of side effects associated with statins, effects on muscle, liver, and in younger individuals, onset of diabetes. So, because of these limitations, we need to look at other avenues for lowering the burden of atherosclerosis and its complications, and there have been some successes. So, for example, this agent ezetimibe targets the 25% cholesterol that's of dietary origin and inhibits absorption of cholesterol at the intestinal level. One particular emerging target is this protease PCSK9. Now, what happens is that when the LDL receptors bind to LDL particles, they undergo endocytosis, and in the lysosomes, the LDL is degraded to give rise to cholesterol, and the receptors are then actually recycled back to the cell surface. What PCSK9 does, it causes modification of the LDL receptors. So instead of being recycled, the receptor undergoes lysosomal degradation, thereby lowering the LDL receptor levels on the cell surface. And so one sort of emerging therapy in the last few years has been monoclonal antibodies against PCSK9 that prevent this modification of the LDL receptor, so they are recycled back to the cell surface. But this is actually quite expensive therapies. Monoclonal antibodies typically cost around $8,000 American per patient per year. So it has to be restricted to those who are extremely high risk. And of course, this will not work in obviously countries like India, where of course you have got a sort of a population factor which is actually at more of a poor end financially. And so if you go and look beyond this, almost a dozen pharmaceutical leads have failed at the clinical level, which has cost billions to the industry. And this is not surprising, because here is a disease where agents have to be taken between 30 to 50 years in the lifespan of an individual. And it's very difficult to predict side effects unless you've got genetic evidence. And PCSK9 is one example where there's genetic evidence. Individuals who have deficiency in PCSK9 don't develop atherosclerosis, even if they've got high plasma cholesterol levels. So that's an example of genetic evidence directing therapies. If you're taking an agent for 30 to 50 years, which has got multiple targets, clearly you're going to have side effects if you're drastically lowering the action of that target. So what beyond manipulating lipid homeostasis, cholesterol levels? Well, of course, we all know that atherosclerosis is an inflammatory disorder, and we know that inflammation is regulated by cytokines. So one particular interesting target is actually sort of cytokines themselves. And indeed, you find a number of pro and inflammatory cytokines which are expressed in atherosclerotic lesions in animal models and also in humans. And what you find is that pro-inflammatory cytokines predominant anti-inflammatory cytokines. So what you can think about is target pro-inflammatory cytokines either by using small molecule inhibitors of the cytokines or key components of its intracellular signaling pathways. You can use neutralizing antibodies that mop up the cytokines. You can use soluble decoy receptors. So these are receptors which are in the solubilized form. They'll bind to the cytokine in the cytoplasm, preventing it from binding to the cell surface receptor. So decoy receptors are small cytokine receptors which mop up the cytokine, preventing it from binding to the cognate cell surface receptor. And also you can think about manipulating cells which produce, manipulating immune cells which produce certain cytokines. So inhibiting those which produce pro-inflammatory cytokine and stimulating those which produce anti-inflammatory cytokines. And indeed, almost, I would say, 10 to 12 years, we have worked on various aspects of cytokines. And what you'll find in terms of references, most of them are predominantly published work from my laboratory. Now and then, in fact, there are references which are outside my laboratories. In this case, Beck and Hansen, in fact, is independent of work in my laboratory. So this can actually act for further reading if required. And so what we tried, because of this attractiveness of cytokines as a sort of therapeutic target, try to look at cytokine actions in more detail. But this is actually not simple. And the reason it's not simple is when you start looking at the number of cytokines and cytokine receptors involved in atherosclerosis and a lot of other diseases as well. So these are chemokines, chemokine receptors, interleukins, and other cytokines with known effects on atherosclerosis, and they're present in atherosclerotic plaques. It's of course not the numbers of cytokines involved. If you look at immune cells, there's a lot of different polarization states of various immune cells, creating complexity, and newer type of immune cells are being identified from more sophisticated single-cell RNA sequencing approaches. So if you take, for example, macrophages, there are at least sort of 4 to 8 different phenotypes, and that's increasing all the time. The same applies with other immune cells, such as T cells. And in terms of the interleukins and cytokines that are shown in red, are some of those on which we have done work in the past, trying to understand their effects in more detail. It's a lot of work spanning about 10 to 12 years. And I'm just going to use one example of a cytokine that we have studied and to just give you the sort of feel about the scale of research activity required. And this is just one cytokine. If you take, in fact, the number of cytokines involved, the list, in fact, goes into hundreds. And the cytokine I'm going to talk about is interleukin 33. And interleukin 33 is a more recently discovered member of the interleukin-1 families. You all have heard about interleukin-1, interleukin 6, and of course, Atrimed works on interleukin 6. Obviously, the interleukin-1 family goes up to interleukin 37, and we've got some research going on at the moment on interleukin 35. Now, interleukin 33, IL-33, a more recently discovered member of the family, it's got 2 cell surface receptors. One, which is the large form of ST2 receptor. So the receptor is called ST2, and ST2L is the large form of the receptor, which is membrane-bound and initiates intracellular actions of the interleukin 33. There's also a small soluble ST2 receptor. So this is in the soluble form. It's a deco receptor which binds IL-33 and mops up the cytokine. And our interest in the interleukin 33 ST2 receptor axis in atherosclerosis emerged from some studies carried out that showed that the soluble ST2 levels were increased in myocardial infarction and heart failure. And there was another study that showed protective effect of interleukin 33 in experimental models of heart failure. So work carried out by our collaborator using the apolipoprotein E deficient mouse model of atherosclerosis went on to demonstrate that if you inject interleukin 33 into these mice, which are fed a high-fat diet, you attenuate atherosclerosis. On the other hand, if you mop up interleukin 33 by injecting this soluble ST2 receptor, you exacerbate the disease. And of course, the studies then went on to identify a mechanism where interleukin 33 causes a change from a pro-inflammatory T helper 1 type of response to an anti-inflammatory T helper 2 type of response. And also, this mice had high level of antibodies against oxidized LDL, which were produced in an interleukin 5 dependent manner. Let me just show you a couple of pieces of data from this published studies. So here, in fact, is obviously the lumen of the arteries, the aortic root of these animals injected with PBS. And here, in fact, is injected with IL-33. And as you can see, that the lumen, in fact, is much larger here. And if you look at the plaque area, what you find is that interleukin 33 causes a reduction in the plaque area. If you look at the immune cells in the plaque, you find that there's a reduction in macrophages and T cells and there's no change in smooth muscle cells or collagen, which is produced by the smooth muscle cells. So here is injecting interleukin-33. If you do the other experiment where you mop up interleukin-33 by injecting the soluble ST2 receptor, which is sort of mops up the interleukin-33, you see a reversal. The plaque area is actually increased. And in this particular case, it works independent of macrophage or T cell levels. But if you look at inflammatory cytokine, particularly interferon gamma, which is a pro-inflammatory cytokine, its levels are increased. While if you look at an anti-inflammatory cytokine, interleukin-5, its levels are decreased. So these were quite interesting results, and we followed it up by looking at macrophage form cells in the plaques of these animals, and we found that they were also reduced. We also went on to look at the mechanisms by which this reduction in form cell formation occurred. And what we found was there was a decrease in uptake of modified LDL by these macrophages, and there was stimulation of efflux of cholesterol from form cells. And so all these studies pointed of IL-33 being protective in atherosclerosis, so a potentially promising therapeutic target. And so when we started doing additional studies in macrophages, we also found a lot of protective effects. So it inhibited the expression of proteases, which cause plaque rupture by thinning the fibrous scarp. It inhibited the expression of a lipase, lipoprotein lipase, which is pro-atherogenic in macrophages. It also inhibited pro-inflammatory genes such as chemokines and adhesion proteins. And so when we started looking at macrophages, wow, there were lots of protective effect. But this is one of the unfortunate situation, or one unfortunate action associated with any cytokine that you look at. Whilst there are protective effects, there are also, in fact, bad effects, detrimental effects. So in other words, we've got a double-edged sword. And so if you look at interleukin-33, some actions of interleukin-33, particularly in endothelial cells, are pro-atherogenic. And this actually applies to this double-edged sword action applies to virtually every cytokine that you can think about. And over a number of years, we have dabbled and looked at the actions of lots of cytokines in relation to form cell formation, of course, a critical early event in the pathogenesis of the disease. And here, in fact, is lots of published papers. Form cell formation is actually a balance between uptake of cholesterol by macrophages, which is bad, and efflux of cholesterol out of macrophages, which is good. And so what we found is that if you look at pro-inflammatory cytokines such as interferon gamma, it inhibits uptake and stimulates efflux, which is really excellent. But again, interferon gamma has some protective effects. If you look at anti-inflammatory cytokines, and I presented some data on interleukin-33, transforming growth factor beta is another one. Because it's anti-inflammatory, it inhibits uptake and stimulates efflux, thereby being protective, but that's in macrophages. There's a paper in Nature Medicine last year which showed that in endothelial cells, TGF-beta is bad. In other words, it's pro-atherogenic. So you got this double-edged sword. And that applies to every cytokine. And so it's not surprising that when you start looking at clinical trials which have tried to manipulate cytokine levels, the results have been mixed. And I'm going to talk about two very large clinical trials that have targeted inflammation. And if you're really interested in sort of understanding this further, there's a nice article by Chan and myself in Future Medicinal Chemistry, which was published this year. And these are extremely large trials. So one of them is called the CANTOS trial, over 10,000 participants. And in this case, they looked at individuals who had previous myocardial infarction. In other words, they were already on statins, but they actually had a high level of inflammation as shown by the high levels of high sensitivity C-reactive protein. And the target here was a monoclonal antibody against interleukin-1 beta, which was one of the first cytokine for which work was done on atherosclerosis. 3 doses were injected. This targets the IL-1 beta, IL-6 pathway. And what was found was that they significantly lowered cardiovascular events. So extremely promising findings. But the individuals were more prone to infection. So that meant that you had to restrict this to high-risk individuals. And of course, there are cost implications as well, because monoclonal antibodies are expensive. To circumvent this, there was another trial with around sort of 5,000 participants, which was based on the rationale that individuals with arthritis are predisposed to atherosclerosis. And what's worked quite well in arthritis is low-dose methotrexate. So in these trials, they took individuals which actually, again, had previous myocardial infarction or cardiovascular event. They gave them low-dose methotrexate as a broad-spectrum anti-inflammatory. No observed benefits were found, and the trial was stopped last year. Again, a huge cost because you're talking around 5,000 participants. And so this actually sort of brought along several sort of points that we have to bear in mind. The double-edged sword, which I mentioned, interleukin-1 beta, while it's pro-atherogenic, there have been some recent studies that have shown that it also stabilizes atherosclerotic plaque. The other thing, of course, is that by targeting inflammation, you're going to make individuals more prone to infection. And also, maybe targeting specific cytokines may be better than using broad-spectrum anti-inflammatory. So the net, what I try to sort of mention is that while there have been some successes in the field of therapies, pharmaceutical therapies on atherosclerosis, there have been numerous failures. And one promising target, inflammation, I've illustrated firstly about the huge number of studies required to understand mechanisms of cytokine actions, but also the mixed results of very large clinical trials. And so that has brought a lot of interest recently on natural products. And of course, natural products have got an excellent safety profile from work which has been carried out over numerous years. And we actually also know the protective effects of natural products from very large studies based on diet and cardiovascular disease. For example, the Mediterranean diet has been found to be protective. So over the last 5 or 6 years, research in my laboratory has moved quite heavily onto natural products. But research on natural products have lagged that on pharmaceuticals on 2 key aspects. One is large clinical trials. And I showed you, in fact, that the scale involved. The Cantos trial, 8,000 participants. 5,000 participants in the third trial. So this is the scale. With natural products, we're talking in a lot of studies which are more 50 to 100 participants, really very low scale. The other thing, of course, is lack of mechanistic insight. And this lack of mechanistic insight is something which we have dabbled and then tried to understand in more detail. And in 2016, we had a really nice, highly cited article in Nature Reviews Cardiology, which dealt with all the natural products in cardiovascular disease at that particular time. And this is one of the figure which is taken from that article. And it shows you that apart from omega-3 fatty acids, our understanding of natural products in terms of mechanistically on the protection against atherosclerosis and cardiovascular disease is limited. And is also worth mentioning is not just one-way traffic, because studies on natural products can give rise to potent pharmaceuticals. And statin, of course, is one example of a natural product, in fact, moving on to more synthetic chemistry. And here, in fact, is another example. So a paper in Cell which showed that some of the actions of omega-3 fatty acids found in fish oil were mediated by a cell surface receptor called G-protein coupled receptor 120. And then a follow-up study where a selective agonist was found of a G-protein coupled receptor 120 that improved insulin resistance and chronic inflammation. So in my laboratory, we have worked on pure natural products and also, in fact, as combinations. Several agents have been studied, polyphenols found in olive oils, omega-3 fatty acids in fish oils, certain beneficial omega-6 fatty acids, flavonols, phytosterols, and then probiotic organisms. And obviously, because of this talk is being recorded, all my results, in fact, is a summary of published work from the laboratory. We've done, of course, a lot of work which has been unpublished, and I'm happy to talk, in fact, that later on, in fact, in Q&A sections, and also, in fact, via further communications later on. Natural product research, of course, got a boom last year by this large trial, REDUCE-IT trial, of 8,000 participants. And the rationale for this study was based on omega-3 fatty acids. Now, some of the earlier sort of studies on omega-3 fatty acid-rich diet and cardiovascular disease were successful because they actually had a higher baseline of intake, because the population was either Mediterranean in origin or Japanese in origin. Then subsequently, using the same concentration, of course, the baseline was lower, so in fact, the overall intake was lower. You had lots of mixed mixed outcomes. So what this was taking into account, so they used a very high dose of an ethyl ester of one of the omega-3 fatty acid, EPA, to mimic that found in Mediterranean population and also Japanese population. And so these were individuals who were on statins, had high triglyceride levels, and they were given twice daily doses of an ethyl ester form of this omega-3 fatty acid, EPA. And what was found was that the level of cardiovascular death was reduced. And there is, in fact, an ongoing trial called an EVAPORATE trial, where they've also seen some reduction in atherosclerosis. Of course, when we're talking about fish oils, we're also talking about depletion of fish, which is, of course, an acute sort of problem. We're also, in fact, looking at environmental issues. So we need to look at other polyunsaturated fatty acid, or PUFAs. And in fact, the general belief that omega-6 fatty acids are bad is not universal. There are some beneficial omega-6 fatty acids, and one of them is dihomo-gamma-linolenic acid, which I'll call DGLA, and I'll present some data on that soon. And then I'll sort of present some studies on probiotics, because they're actually sort of quite cheap and perhaps more applicable to the Indian population. So here, in fact, is some studies, in fact, on omega-6 fatty acid, DGLA. As I mentioned, that there have been some previous studies that have shown that it's anti-inflammatory. And DGLA is synthesized from gamma-linolenic acid, or GLA. And you find GLA even in primrose oil and borage oil, of course, which are some supplements which are taken in certain parts of the world. Now, up to recently, our understanding of DGLA and atherosclerosis was quite limited. It's also worth mentioning is that these studies were carried out in collaboration with Ben Gurion University in Israel, who were produced in a mutated algae that produced DGLA as 20% of their dry weight. Now, what got us interested in DGLA was some studies carried out by our collaborator using the apolipoprotein E deficient mouse, which have been fed a high-fat diet along with DGLA. And what these studies found was that when you looked at the aorta, there was reduced levels of lipids. There were also reduced inflammation because there were reduced level of macrophages, also reduced level of adhesion proteins, which are associated with inflammation. There was one particularly bad effect, which was there was also reduced level of smooth muscle cells, among lots of good effects. But what these studies didn't indicate was mechanisms. How do you explain these changes that you see in the animal models mechanistically at the molecular level? And this is what we went on to carry it out. So, for example, our preclinical studies showed that there was reduced plaque macrophage content. And we went on to show that this was because DGLA inhibited chemokine-driven monocytic migration. The preclinical in vivo studies showed this reduced plaque inflammation. And we went on to show that DGLA inhibited pro-inflammatory gene expression in macrophages by 3 pro-atherogenic cytokines: interferon gamma, interleukin-1 beta, and tumor necrosis factor alpha. And in the case of interferon gamma, we actually pinpointed the mechanism to a single phosphorylation change in a key transcription factor called STAT1, or signal transducer and activator of transcription 1, and DGLA inhibited interferon gamma mediated phosphorylation of this transcription factor on serine 727. And when we looked at over 80 genes, we found that those involved in inflammation, adhesion proteins, and also those involved in lipid homeostasis were affected. We also could explain why there reduced smooth muscle cell content in the animal model systems, because DGLA inhibited the migration of smooth muscle cells, but also inhibited proliferation of endothelial cells. What about reduced levels of plaque lipids or lipids in the aorta? And so what we did here is we took some macrophages, we incubated them with acetylated LDL, which is like oxidized LDL taken up by macrophages and which converts into form cells, either in the presence of vehicle or DGLA, or in fact with vehicle in the absence of DGLA. And in these experiments, we actually had 14C level acetate, so radioisotope of cholesterol, of carbon in acetate. Now, one of the hallmark of form cells is that they have increased intracellular level of cholesterol esters. And so if you look at this highlighted area, in the presence of just acetylated LDL, you've got a massive increase in the cholesterol ester level, and this is inhibited by DGLA. So DGLA inhibits macrophage form cell formation, which then accounts for the reduced level of lipids that we see in the aorta. Now, of course, when I actually talked about cholesterol and form cell formation, I mentioned, in fact, it's a balance between uptake, which is part of so-called forward cholesterol transport, which is bad. So this is where macrophages can take up LDL or modified LDL, either by receptor-mediated endocytosis or by other processes such as macropinocytosis or phagocytosis. And when you look at DGLA and the effects on these individual processes, we find that it causes inhibition. So here, in fact, is DGLA-mediated inhibition of receptor-mediated endocytosis. So we used oxidized LDL, which is fluorescently labeled. And here is DGLA-mediated inhibition of macropinocytosis, where we use a fluorescently labeled dye as a marker of macropinocytosis. We also went on to show by a variety of assays that DGLA also inhibits the expression of scavenger receptors, which are involved in this receptor-mediated endocytosis. And here are two scavenger receptor A and CD36, and the expression being inhibited at both the messenger RNA and also at the protein level. So that is uptake of cholesterol, which is bad. There's also a process called efflux of cholesterol, which is good. So in this case, the cholesterol from formed cells is picked up by HDL, high-density lipoprotein, which is, of course, a good cholesterol carried by your body. The HDL will then take it to the liver, and the liver, the cholesterol will be converted into bile. And we know from our sort of school days, lessons on, in fact, on digestion, that bile emulsifies fats and reduces their surface area for the action of lipases. And indeed, that's one of the job of bile produced by the liver, and of course then stored in obviously the gallbladder and secreted, in fact, during the digestion process. 95% of this bile is absorbed back, 5% you get rid from the body, and this is the route, the only route by which the body can get rid of cholesterol. That's why this reverse cholesterol transport and cholesterol efflux is a good process. And when you look at DGLA in macrophages, it stimulates cholesterol efflux. So DGLA is synthesized from GLA. And GLA, as I mentioned, is found in evening primrose oil and borage oil. So it's not surprising that GLA is also anti-atherogenic. And here are some sort of data. It inhibits interferon gamma-induced pro-inflammatory gene expression and also chemokine-driven monocytic migration. GLA is not as potent as DGLA in its action. So the next question we ask is that's fine. What are the intracellular mediators of DGLA actions? What happens once DGLA is taken by the cells? What does it get metabolized? And which metabolites carry out the actions of DGLA? So here we went on and carried out what was called a mass spectrometric based approach. And we found that the levels of one of the icosanoid, prostaglandin E1, was increased by DGLA under inflammatory conditions when you interferon gamma, or in the non-inflammatory condition, just DGLA, a massive increase. And this correlated with actions of DGLA also being also seen when you use PGE1. So instead of DGLA, if you add PGE1 to macrophages, prostaglandin E1, you see similar responses, such as interferon gamma-induced pro-inflammatory gene expression and chemokine-driven monocytic migration. You can also knock down the expression of enzymes which are involved in the formation of prostaglandin E1. And the enzymes involved are cyclooxygenase 1 and 2. And you can knock down the expression of cyclooxygenase 1 and 2 using small interfering RNA. And this is what we have done here. So here we're looking at interferon gamma-induced expression of this MCP1. And if you look at our negative control, DGLA still inhibits this induction. So you see this reduction by DGLA. However, if you knock down the expression of cyclooxygenase 1 and 2, and thereby the production of prostaglandin E1, you no longer see this reduction. So that actually sort of goes on to point that PGE1 is a mediator, central mediator of the actions of DGLA. And you actually sort of carried out a lot of other work, which I'm not going to talk today. This is, of course, unpublished work. Now, the last story that I'm going to tell you about, and again, what I'm going to talk about is published work. We've also done a lot of unpublished work, which is actually being written up at the moment. This is something obviously interest in India. It actually sort of came about in part of this study published in Nature on sepsis in a sort of a trial in rural India, where there was a randomized symbiotic trial which prevented sepsis. And one of the key agents here was Lactobacillus plantarum. And of course, Lactobacillus plantarum is a probiotic bacteria. And of course, probiotics are relatively cheap. And so that brought us an interest in potential probiotic as anti-atherogenic preventatives, and also potentially being used as therapeutics with pharmaceutical agents. And we all know that the gut microbiota plays a key role in the various pathologies, including atherosclerosis. So if an individual has got a diet which is rich in red meat, then you've got high levels of phosphatidylcholine and L-carnitine, which the gut microbiota will convert into trimethylamine. The trimethylamine will then be converted into trimethylamine oxide, which then mediates pro-atherogenic effects, such as it reduces reverse cholesterol transport and stimulates forward cholesterol transport, thereby causing atherosclerosis. And probiotics have got many beneficial effects in relation to atherosclerosis. And here's a diagram taken from one of our articles recently published in Molecular Nutrition and Food Research that shows you the beneficial effects of probiotic bacteria. And again, I would highly recommend that you read this if you're interested in probiotics. So the research, in fact, in probiotics came along because of a collaboration with a local probiotic company called Caltech Limited. Our sort of previous collaborative study had shown that Lactobacillus plantarum Cal66, a probiotic bacteria, cause lowering of cholesterol in the CACO2 enterocyte cell culture model system. Some other published studies by the company also showed that a so-called Lab4 consortium of probiotic, which contained 2 species of Lactobacillus acidophilus and 2 species of Bifidobacterium bifidum, also had some anti-inflammatory activities. So the question we asked was that what when you combine the 2 probiotic mixture and look at its effect? And the study which I'm going to describe only looks at the sort of risk factors associated with atherosclerosis in mice which have been fed a high-fat diet for a short-term duration. We, of course, went along and have done studies on atherosclerosis, which are unpublished. And also the company has done some trial on 400 individuals where they've shown some successes with some of the parameters that we've also seen in our preclinical studies that I'm going to present in a minute. So this is our experimental design. 18 wild-type mice. 6 were fed a chow diet for 2 weeks. This was our baseline. The other 12 were split into 2 groups. 6 fed a high-fat diet containing cholesterol with no probiotics, and the other 6 fed a high-fat diet with probiotics. And then we looked at a number of parameters, lipids in the plasma, cytokines, and we also looked at metabolites in tissues, and also looked at gene expression. So here are some summaries. First of all, if you look at weight gain by high-fat diet, what you find is that the probiotic significantly inhibits this in this model system. And of course, the companies went on to show that this also occurs in 400 participants in an article published in Scientific Reports last year. If you look at plasma lipids, so this is total cholesterol, VLDL, LDL cholesterol, and HDL cholesterol, and they got triglycerides. Compared to baseline, high-fat diet causes an increase in total cholesterol, VLDL, LDL cholesterol, and also HDL cholesterol. If you look at probiotics, there's a significant reduction in total cholesterol, and also significant reduction in LDL, VLDL cholesterol. There's no change on inflammation markers. So the next question is that what is responsible for this reduction in total and LDL, VLDL cholesterol that we see in these animals. As I mentioned, is that the only way the body can get rid of cholesterol is via the bile system, which we use about 5% of bile. And this is the pathway involved. So cholesterol is converted into bile by this rate-limiting enzyme, which is abbreviated as CYP7A1. So that's the rate-limiting enzyme in the biosynthesis of biosynthesis of bile in the liver. So cholesterol into bile, the rate-limiting enzyme is the CYP7A1. So we looked at the bile levels in the plasma and also in the fecal and the feces of these animals by a sort of mass spectrometry-based approaches. Here we've got a principal component analysis. Here we've got heat maps. And as a summary, if you look at the plasma, there was actually no change in bile acid. But if you look at the fecal material, you found that there was significantly increased bile levels. So there was increased excretion of bile. And this you saw total or unconjugated bile acids, they were always significantly higher in the probiotic group. You can also look at individual bile acids in the feces of these animals. Again, increased levels of individual bile acids, also deoxycholic acid, higher deoxycholic acid. So there was increased bile which was got rid from the body in the feces. So how could we explain this? What are the mechanisms involved? So for this, we started looking at gene expression. We looked at gene expression in the liver. We looked at gene expression in the intestine. And what we found was that there was changes in gene expression in relation to bile acid metabolism. And so if you look at the rate-limiting enzyme in the synthesis of bile in the liver, CYP7A1 or cholesterol 7-alpha hydroxylase, its expression was increased. So that means there was more bile being produced by the probiotics in the liver of the animals. So this is a rate-limiting enzyme involved in the synthesis of bile. There's also an inhibitor of the transcription of this enzyme, and this inhibitor is called a small heterodimer partner, with a transcriptional repressor of CYP7A1. And consistent with the inhibitory effect of this on CYP7A1, we found that its level were reduced in the liver of animals which were fed probiotic compared to just high-fat diet. So we could pinpoint the mechanisms for the changes that we saw in the plasma cholesterol levels, which was increased bile synthesis in the liver because of changes in the expression of key rate-limiting enzymes and also transcription factors which are involved in the production of bile, and therefore increased excretion of the bile from the body. So I hope, in fact, I've given you a flavor by these two studies that atherosclerosis is an inflammatory disorder where cytokines play a key role in all stages of the disease. Current therapies I've talked about, statins, ezetimibe, PCSK9 inhibitors. I've also, in fact, talked about cytokine therapeutics and the mixed result. But one point to bear in mind is that the current therapies, so the statins, are associated with residual cardiovascular risk, together with various effects. And so we need to look at alternatives. While in alternatives we've got some successes, there have been lots and lots of failures. And the failures goes, in fact, into dozens. So we need to now look at natural products, but we need, in fact, in terms of natural products, deeper mechanistic insight and large clinical trials. And so I've gone on to show you that at least with two agents that we are studying, the omega-6 fatty acid DGLA and probiotics, we actually know that they inhibit several pro-atherogenic processes in vitro and in vivo. We also, in fact, informed on the mechanisms which are underlying the beneficial actions of DGLN probiotics. And in some cases, the probiotics, in fact, is moved, in fact, to humans as well. And my final slide, of course, is acknowledging our funders, British Heart Foundation. They've funded my research for over 20 years. Also, the local probiotic company, Caltech Limited, Ben Gurion University in Israel. Here, in fact, is a list of my collaborators. And here, of course, some of the people who carried out the work. So the DGLA work was predominantly by Haley Gallagher, a PhD student in the laboratory. And then, of course, some of the work has been taken to the next stage by Jessica Williams, Ala Ismail, and Yu Hong Chang. I had no time to talk about their work. Yi Chang has also worked on other sort of natural products as well. And the probiotic studies were carried out by a PhD student, Victoria Omorian. So I think I'll stop there. Thank you for listening. Hope, in fact, I'll give you a flavor about the field of atherosclerosis, targets, and also the huge potential that natural products have in the prevention and treatment of this disease. Thank you, and I'll stop there. And I'll also, in fact, stop sharing my slides. I can find out.
You can keep the slides on because maybe that we will
Sure.
go back.
Yeah, sure. Yeah, that's fine. Yeah.
Yeah, Bharat, any questions?
Yes, sir. Thank you, sir. It was really a very informative talk, and I would like to take a couple of questions from audience side. Very first question I would like to take is regarding the animal model being used in the atherosclerosis study. A question goes like this: majority of drugs, including statins, are developed using only one genetic model, that is C57 mice, and used by all people across the world. Is it could be the reason for major unwanted effects caused by these statins?
Well, I think there are two things here, right? Is translation of animal model studies into humans, okay? And that, of course, is a major hurdle, right? So C57 BL6 mice, right, is actually not a model for atherosclerosis, because even if you've kept feeding these mice a high-fat diet, they're not going to develop much atherosclerotic. But this is a good model for looking at metabolic perturbations, right? Metabolic syndrome and risk factors associated with the disease. You need to go into, obviously, the two mouse models that I described, the LDL receptor deficient mouse model and the apolipoprotein E deficient mouse model, which have to be fed a high-fat diet. Again, of course, each model has got their limitation. And of course, in some countries, the rabbits have been sort of utilized as well, and non-human primates. Clearly, one has to actually be cautious in terms of extrapolating, because animal models are different from humans. In fact, in a lot of aspects, I mean, two key aspects is lipid homeostasis. So mice carry HDL as their cholesterol. Humans, in fact, carry cholesterol as LDL particles. And of course, there are differences in the immune system between mice and humans as well. So there is one of the hurdle. Then, of course, the other point to bear in, so, we are trying to develop new model systems. So one particular model system is using wild-type mice, so there's no genetic deficiency, but you actually inject into them an adeno-associated virus against this protease PCSK9 that I mentioned. So this is actually away from the genetic background. But again, that model has got sort of various limitations. So that's one thing to bear in mind. But, we have to start at some point. And if you look at statins, the work on statins originally was actually carried out in cells. And it was not even, in fact, a human cell. They were carried out in yeast cells, looking at cholesterol sort of metabolism in yeast cells. Then it led to human cells. Then, of course, animal models. And then, of course, eventually you need large human trials. Worth bearing in mind that we've got a very heterogeneous worldwide population that's not often reflected in sort of clinical trials. So what you actually find in a large clinical trial on Caucasians will not be applicable to, say, the Indian population. So that's why, in fact, you have got these issues, in fact, where a tried and trusted sort of agent in one clinical trial will actually produce various sort of issues. And one of the things when, of course, statins was extremely successful was people thought, well, let's give them to a younger population. And they found that it caused sort of diabetes. And that's because HMG-CoA reductase and cholesterol is quite an essential component, right? Cholesterol is required for membranes, is required for sort of brain physiology and so on. So you need to be careful about, prescribing this, in fact, on a universal basis. So that's why, in fact, you have some mixed sort of results. So I wouldn't say, in fact, that is. I know, in fact, that wild type mice are used universally. I don't think, in fact, that that's the reason for the issues with statins and other pharmaceuticals. Mice, of course, are useful animal models because of the genetics. But in terms of identifying agents, we need to actually sort of carry out clinical trials. And that clinical trials, in fact, has to be multinational and take the whole heterogeneity in the various population into account to actually get a full effect. So I've, in fact, in a sort of more rounded manner, tried to describe sort of quite key limitations that we have. But in we are moving somewhere in terms of, but we need to have more clinical trials, and of course, involvement of countries like India. Okay. Have I?
Yes, sir.
Okay.
Thank you, sir. I feel the question is very well answered. And I would like to take the next question, and it is regarding there is a lot of data available on preclinical investigation of herbs and natural products on atherosclerosis and its validation, and how we can evaluate it based on the personalized intervention in clinical trials.
One thing is worth bearing in mind, right? That diet, obviously dietary components, right? When you start giving it to individuals, you have to be extremely careful if individuals are taking pharmaceuticals. And so sometimes, when we're watching, in fact, sort of various media things, and sort of channels about herbal sort of therapies, in fact, and so on and so forth, if it's actually taken in the wrong way, in fact, it can cause sort of more harm. And let me just give you one example, right? Grapefruit juice. Grapefruit juice, in fact, is wonderful, right? And in the West, in fact, people drink that, right? But if you're actually taking statins, it's actually bad news, right? Because you're going to affect the metabolism of statins, right? So, here is, in fact, an interaction that you see between sort of a food product or food agent and, of course, a pharmaceutical. So, those things, in fact, have to be taken into account, right, when you're looking at the benefits. 2 points. One, in fact, is prevention. And that's fine. In terms of a lot of these natural products, prevention, you don't have the issues, the concerns that I sort of mentioned. And I think diet, exercise, and of course, these agents, in fact, as preventatives, is actually a sort of way we need to move forward. But when you just start looking at therapeutics, then, of course, you need to be careful because a diet, sort of what is actually good at prevention may not cause regression of atherosclerotic plaques in individuals who already got atherosclerosis. And also you need to look at the interaction of your herbal remedy or whatever natural product with the pharmaceuticals that the individuals are taking. And so for that, I think two critical requirements. One is actually large clinical trials. That's the problem with natural products because there's not much money, in fact, as far as big pharmas are concerned. And reduce it is one example which actually went along and showed those mixed fluctuation in sort of results which we're getting from small clinical trials in more sort of single population when they took everything in account and realized, well, we needed a much higher dose of omega-3 fatty acid, and they developed an ethyl ester, which was then actually tried in an extremely large trial of 4,000 individuals, right? And that actually sort of showed that yes, in certain individuals who are actually on certain pharmaceuticals, yes, this is actually a sort of a viable option. So I think there is a lot of studies coming out on herbs and natural products, working on cell culture model systems, and sort of coming out with the tag that, well, this is going to be effective, maybe in fact on sort of wild type mice. I think you need to actually expand that. You need to understand it in more detail. You need to actually sort of then eventually carry out large clinical trials and then actually see what the outcome may be. Which of course is a lot of money, but you can also look at certain themes in diet, right? So a lot of early studies, in fact, on diet, Mediterranean diet, Japanese diet, were actually sort of more survey-based, and so you can actually glean a lot from those type of studies as well. So there's actually no simple answer to that question, right? It's that we need to understand it in more detail, sort of things which we need to carry out, but we need to also be cautious by sort of saying, oh, well, look, this actually shows some success, go along and take that, right? Because if you're actually on pharmaceuticals, then you may actually have interactions. Another point to bear in mind is that this is a dynamic process. So you should not actually extrapolate even in clinical trials too much back, say 10 years ago. Because 10 years ago, you only had a small proportion of world population on statins. Now you've got a much larger population which is on statins, and they've got much more stabilized plaques compared to what they were 10 years ago. So if you find something which was successful in clinical trials 10 years ago, may no longer be the case now, because in fact statins have stabilized their plaques. So you have to be careful about in terms of when the studies are carried out as well. But in the sort of short, more studies, more larger clinical trials, and not in fact on sort of a smaller scale. In fact, I've now sort of come across and talked with various visits out in India. In fact, 20 sort of individuals have been taken, and sort of product, in fact, sort of mentioned that this is actually sort of effective, and that's actually sort of okay. In fact, in terms of sort of convincing, in fact, a population which is perhaps not well. Of course, the education is increasing, but I think you need to actually have sort of more robust, peer-reviewed large trials, which of course needs a lot of money.
Yes, thank you very much for your detailed answer. And I have a next question regarding your DGLA study. In your DGLA study, did you conduct long-term studies, or the effect is for short-term only?
Okay. So DGLA studies, in fact, we have done short-term studies, but the studies that I described, right, was actually one by our collaborator, which was actually on a mouse model of atherosclerosis, the apolipoprotein E deficient mouse model. That was actually a long-term study. And there's also, in fact, studies which we have carried out, which are actually long-term studies as well. So this is actually sort of talking about 12 to 24 weeks of high-fat diet.
Thank you very much, sir. And during your presentation, you have also mentioned regarding the microbiome and atherosclerosis. Can you please elaborate on microbiome and its relation in immunity?
Okay, I mean, that's really a sort of not a simple sort of question to sort of to address, because it's extremely complex, and we're actually sort of learning a huge amount, right, about changes in microbiome, right, from changes in our diet and changes, in fact, that actually sort of occur in various diseases. And if I just sort of go along, and one of the things, in fact, which just try to sort of maybe, in fact, this is the only way I can sort of perhaps go back. Oh, yeah, okay. So if you go on this particular slide, this slide is just actually looking at microbiome in relation to atherosclerosis and how, in fact, it affects immune system and inflammatory responses just associated with this disease, right? And so this is just one example. I recommend that you read this article if you want to have more detail. So here, in fact, is the number of effects which with the microbiome may have, if, for example, if you got a high-fat diet, it will cause leakiness of the gut, which will, of course, imply that the LPS will then actually move from the gut into the plasma. That will actually cause increase in pro-inflammatory cytokine signaling and so on. Also nitric oxide mediated vasodilation. Of course, the arrows in this case are going down because I'm showing the actions of probiotic. But if probiotics were not present, clearly one example would be leakiness of the gut, which would cause lipopolysaccharide to leak out, causing increase in toll-like receptor 4 signaling, causing increased production of pro-inflammatory cytokines. It will also, in fact, cause changes, in fact, in vasodilation. We also, in fact, have increased production, as I mentioned in the previous slide, of TMAO. So if you go to high diet, which is rich in red meat, the gut microbiota will produce TMA, which will be converted into TMAO by the liver. And then this TMAO will also go on, in fact, and increase vascular inflammation, which, of course, then is reduced by probiotics. So it shows you, in fact, that there are actually multiple mechanisms. And here, in fact, I've just actually mentioned two potential mechanisms. One is, in fact, leakiness of the gut by detrimental microbiome, which will cause pro-inflammatory agents such as lipopolysaccharide to move into the plasma and then affect immune cells, but also metabolites of the bacteria under dietary setting. So in this case, you got sort of the L-carnitine converted, in fact, eventually into TMAO, which then also, in fact, has a direct effect on cells in the vasculature and also immune cells. So, you know, there are actually sort of no single mechanism. There are multiple mechanisms. I'm just illustrating two here, in fact, in relation to atherosclerosis.
Thank you very much, sir. And I have a next question. With your experience in the field of cardiovascular disorders, would it be possible to explain the relationship between xanthelasma pulperbum and cardiovascular disorders?
Yeah. So one of the point which is worth bearing in mind is this thing about comorbidities, right? In the sense that, you know, cardiovascular disease, atherosclerosis, you know, obviously changes happening in the arteries and the clinical complications, of course, which are associated with the arteries, so plaque rupturing and then blocking the artery is obviously one aspect that we're looking at. There's, of course, inflammation associated with a lot of other sites, which eventually feeds in either by changing the risk factors associated with cardiovascular disease or the immune cell profile in either the plasma or the arteries or, of course, in the bone marrow, and then impacting atherosclerosis. So to give you sort of some examples, if you go back to my earlier sort of point about the CERT trial, which I mentioned, which was not successful, that was high incidence of cardiovascular disease in individuals with rheumatoid arthritis. And so where low-dose methotrexate was successful and therefore used, in fact, in individuals in cardiovascular disease. So that's one link, an inflammatory disorder, which is actually the arthritis predisposing to atherosclerosis. You take any inflammatory disorder, you're going to have sort of increased cardiovascular morbidity and mortality. Inflammatory disorders, in fact, in the intestine, inflammatory disorder in the skin. So that's one thing. Inflammation, adipose tissue inflammation affecting cardiovascular disease. So psoriasis, inflammation of the skin. There's a lot of evidence, in fact, and we try to get some sort of studies actually carried out here by an expert, in fact, who was in psoriasis, to try and link with cardiovascular disease. So that's inflammation, any inflammatory disorder leading to cardiovascular disease, and then metabolic perturbations, right? So changes, in fact, in levels of cholesterol metabolism, fatty acid metabolism, glucose homeostasis, insulin resistance, they all eventually impact cardiovascular complications. So any agent, any sort of disorder associated with changes in immune and inflammatory responses and metabolic perturbations will actually impact cardiovascular disease.
Thank you very much, sir. And this is all regarding the questions from audience. And now I would like to request our panel members, if you have any doubts, you are free to answer the question.
Yeah, sure.
Dr. Shiban, can you come in?
I'm here. Am I?
Yes, sir.
Can you hear me? Thank
Yes,
you. Thank you, Dr. Ramji. It was wonderful. It's extraordinary in its depth and its breadth. In fact, by the last slide, it was so overwhelming. Too much information in such a little time. I really enjoyed it. Intriguing. I had tons of questions, but when we meet in person, maybe I'll ask you instead of holding off everybody else. Thank you, Atramed. Thank you, Dr. Damlaji. And thank you, all the panel members and Ayurvedic Association members too. And thanks all the participants. This will be available on YouTube later, and you're all welcome to watch it, those who missed it. And you should all join us for our next lecture. We'll announce it pretty soon. Thank you all for joining us. Anything else, Dr. Damlaji?
Yeah, there could be more questions from panelists, so we will let the panelists ask questions.
Okay, sure.
Dr. Manoj, Sadat?
Let me ask one.
Shaidu.
Oh, sorry. Go ahead, please, sir. Please, go ahead.
Dr.
Any
Shaidu?
more questions?
Dr. Shiban, you can ask the question.
Only one question out of my 200 questions. I'll ask only one question. Is atherosclerosis an inflammatory disease or just a mere cholesterol deposit metabolic disease?
Can you repeat that again? I didn't catch it.
Is atherosclerosis an inflammatory disease or a metabolic disease where cholesterol is hyped up a bit too much?
There is no doubt, or the evidence, accumulating evidence in the last 15 years, that atherosclerosis is an inflammatory disorder. In fact, when it comes to undergraduate teaching, where I give them a lecture, in fact, and about the evidence which actually sort of goes along to show that atherosclerosis is an inflammatory disorder. So I think when in the sort of Brown and Goldstein, when they discovered the LDL receptor, they sort of felt that, well, it's actually cholesterol is the culprit. But we now know, in fact, that cholesterol and all the sort of perturbation, dysfunction in lipid homeostasis that you see ultimately act through inflammation and inflammatory cytokines. So cholesterol crystals, for example, activate inflammasome. So although, in fact, the triggers, the risk factors, in fact, are sort of lipids and then sort of hypertension and glucose and so on, the ultimate action is at the modulation of inflammation. So the evidence, in fact, is so strong. And then I think the final evidence, which was the CANTOS trial, which was the large trial with 8,000 individuals, and that actually sort of went along and showed that all of those years of evidence which we had from animal models and looking at inflammatory cells in plaques in humans, well, we can now, in fact, target as a key cytokine and actually lower, in fact, cardiovascular disease. So, that is actually sort of, I think, the ultimate sort of final proof, in fact, on atherosclerosis being an inflammatory disorder. Of course, that inflammation is triggered by a lot of agents, And of course, dysfunctional lipid homeostasis is one of them.
Thank you. Anything else, Dr. Damleji?
I think no more questions. I again personally want to thank Dr. Ramji for his excellent lecture. No wonder it is professor.
I think.
Sir, I have a question, Damle.
Oh, okay.
Yeah, sure. Go on.
Hello.
Yeah.
Hello.
Mm-hmm.
I do have a question.
Yeah. Go on.
So, whether you have worked in any of the plant molecules in atherosclerosis, have you found any? Have you done any work on plant molecules in this?
Yeah. So
atherosclerosis.
Yeah. So I mentioned, in fact, it's a study, in fact, of the probiotic. Of course, is more coming from bacteria, but DGLA, of course, is derived from GLA. When GLA is found from evening primrose oil and borage oil, so that's actually one sort of molecule of plant origin. Then we've got resveratrol is another agent that we are studying, which is actually found in grapes of the skin of the grapes. Putting collagen, pomegranate, so there are a lot of different sort of plant agents that we're sort of studying, in fact, and trying to get more deeper mechanistic insight. The majority of this sort of agents tend to be of plant origin in any case, right? And that's why I sort of feel, in fact, that the Ayurveda, right, has got a lot of potential, right? It just needs a lot of funding, I think, from the Indian government, in fact, and more large trials, more mechanistic insight. But, the majority of these successes have been sort of mainly initially, in fact, from plant origin. Hope that answers that question.
Or any question?
The other point I had to bear in mind, of course, because of the recording, in fact, obviously I can't present all the unpublished data, in fact, to sort of protect people who have done the work, because obviously publication takes quite a substantial time, in fact, from writing. So if they actually wants to talk in this more detail, right, just contact me, and I'm sort of more than happy to spend hours to sort of engage in scientific discussion, right? And as I say, in fact, that to me it was great to actually sort of meet Rishikesh and Lataben, in fact, in Bangalore. I mean, not only we had scientific exchange which led to this, but also, as I mentioned, something about my community service, in fact, and one of the things was Samu Pratna, where in fact Lataben, in fact, sang 2 bhajans, and the audience went, wow, so clearly, in fact, there is, it actually just goes beyond, in fact, our scientific discussion. In fact, goes to our deep culture and heritage, right? And so, it's been sort of a good thing, in fact, wonderful thing to have met both of you, in fact, and I'll be happy to, in fact, be involved in further sort of presentations in the future, not only with Atrimed, but any other sort of organizations. Discussing science, and that's so good, right? Along with our culture and heritage.
Thank you very much.
Thank you, sir. Thank you very much.
Okay. Thank you. Anything else?
Any other questions?
Yeah, thank you all for your gracious presence, and I request all the participants to submit your valuable feedback on a given link on the screen. Please do submit the feedback and get your certificates within 24 hours after the submission of your feedback. Thank you very much. Thank you very much for everyone.
I have an announcement. The next lecture will be how to find toxicity of food and drugs. That's by Dr. Subramania Vangala.
On 19th, no, sir?
Yeah, next meeting.
He's from Bangalore as well, right? I met him in Bangalore. Yeah. And you had Siv Balashankar, in fact, who gave us a talk, in fact, a couple of weeks ago. And he's also in Bangalore. In fact, he's been in Cardiff. And I actually met both of them in Bangalore. So that's so Bangalore, because I said, a place, in fact, to meet sort of scientists, I suspect, and sort of have sort of connections as well.
Yeah, Shiva Balasubramaniam was talking to you. I mean, talking about you when I met him day before yesterday.
Yeah.
So he was having another meeting today. Otherwise, he wanted to attend your lecture. But
Yeah,
next
yeah.
time I'll call him.
Yeah. Great.
Okay, sir. Thank you very much. So let's conclude the day. Again, personal thanks to you. I'll keep in touch with you.
Okay, that's good. Thank you. Pleasure. And
Bye-bye.
thanks, in fact, for hosting me. In fact, it was a really great pleasure having all these discussions. And I hope, in fact, that this webinars that you're arranging, in fact, will increase, in fact, interactions, in fact, and sort of increase knowledge, in fact, in the scientific community, not only in India, but also worldwide. So thanks very much. It was a pleasure. Thank you. Okay, bye.
Bye.
Bye, sir.
Bye.
Hello.